Good morning, everyone. So today we will explore the nervous system. And I, Mary Patricia Tria, together with my group mates, will discuss its anatomy and physiology, as well as how this system contributes to the maintenance of the homeostasis. So first, I will discuss the overview of the nervous system, its organization, and its functions. Then we will proceed to the histology of the nervous tissue, how the electrical signals or nerve impulses travels between neurons and through neurotransmitters. So first, what is nervous system? Well, nervous system is one of the smallest and yet most complex of the 11 body systems. Imagine it is only about 2 kilograms and 3% of our body. It processes many different kinds of incoming sensory information, source of thoughts, emotions, and memories. So when we say neurology, it deals with normal functioning and disorders of the nervous system, while neurologist is a physician who diagnoses and treats disorder of the nervous system. So if you have any problem with your brain, your spinal cord, or with your nerves or neurons, then you should find a neurologist. So this is the organization of the nervous system. If you could see, there are a lot of subdivisions under the nervous system. First, the nervous system is divided into central nervous system and the peripheral nervous system. Our central nervous system contains our brain and our spinal cord, while our peripheral nervous system consists of our sensory division and motor division, while the motor division is further divided into autonomic and somatic divisions. Then, our autonomic nervous system also has sympathetic and parasympathetic divisions. Well, this could have a lot of subdivisions, but we will further discuss this as we will dwell on the topics later on. So first is the central nervous system. Our central nervous system consists of our brain and our spinal cord. Why is it called central nervous system is because most of the signals that stimulate muscles to contract and glands to secrete are originated in here. Thus, it is called central nervous system because almost all of the signals that goes into our body originates in our central nervous system. In fact, our brain contains about 85 billion neurons and our spinal cord has about 100 million neurons. While, on the other hand, we have our peripheral nervous system, which consists of all nervous tissue outside the central nervous system. Well, this peripheral nervous system includes nervous tissues throughout our body, aside from our brain and our spinal cord. Well, there are different nervous tissues that could be found in our peripheral nervous system. It could be a nerve, which are bundle of hundreds to thousands of axons, plus associated connective tissue and blood vessels. The second one is ganglia, which are small masses of nervous tissue consisting primarily of neuron cell bodies, closely associated with cranial and spinal nerves. The third one is the enteric plexuses, which is extensive networks of neurons located in the walls of organs of the gastrointestinal tract. And lastly is the sensory receptor, which is a structure of the nervous system that monitors changes in the external or internal environment. Well, these four kinds of nervous tissues that could be found in peripheral nervous system would be further discussed later on. So our peripheral nervous system also has its division. It is divided into two, which are the somatic nervous system and the autonomic nervous system. So first is the somatic nervous system. When we say somatic nervous system, it has sensory neurons that convey information to the CNS or central nervous system coming from the somatic receptors. And it also has motor neurons that conduct impulses from the CNS to skeletal muscles only. That is why it is voluntary. While autonomic nervous system has sensory neurons that convey information to the central nervous system coming from the autonomic sensory receptors, while its motor neurons conduct nerve impulses from the CNS to smooth muscles, cardiac muscles, and glands, which are specifically involuntary. If you will observe, these two kinds or two divisions of the peripheral nervous system are divided into voluntary and involuntary. Our somatic nervous system are basically the voluntary one. It conveys information or sends signal from somatic receptors, then it produces motor or action potentials within the skeletal muscles only, which are highly voluntary. However, on the autonomic nervous system, from the word itself, autonomic, which is somehow automatic, because those motor neurons that are under the autonomic nervous system are responsible for the smooth muscle, cardiac muscle, and glands, which are highly involuntary. So, this autonomic nervous system is further divided into three. It is the sympathetic division, the parasympathetic division, and the enteric nervous system, or the enteric division. If you will see, the enteric division is somehow isolated from the sympathetic and parasympathetic division. It is because the sympathetic and parasympathetic division are primarily focused on the smooth muscle, cardiac muscle, and glands. However, the enteric nervous system or the enteric division, which is also under the autonomic nervous system, is specifically focused only for the gastrointestinal tract, in which its sensory neurons of the ENS or enteric nervous system monitors chemical changes within the gastrointestinal tract as well as the stretching of its walls. And its motor neurons govern the contractions of the gastrointestinal tract smooth muscle. So that is why it is isolated from the sympathetic and parasympathetic division, because enteric nervous system or enteric division is somehow specialized because it is only focused on the gastrointestinal tract. So if you will see this diagram, you will understand, or the summary, you will see the summary of the whole nervous system. So from our central nervous system, which contains our brain and spinal cord, then we have here our peripheral nervous system, which contains the other nervous tissues or nerve cells throughout our body. Then through peripheral nervous system, we have here the sensory division, which inputs information or sensory towards the central nervous system, and we have here the motor division, which receives information or motor output coming from the central nervous system. And from that motor division, we have the somatic and autonomic. The somatic, which focuses only on the skeletal muscles, and the autonomic nervous system, which is further divided into three: the sympathetic, the parasympathetic, and the enteric nervous system. So now, what are the functions of this nervous system? Well, there are three main functions of the nervous system. First is the sensory function, the integrative function, and the motor function. The sensory function is achieved through sensory receptors which detects internal stimuli, such as an increase in blood pressure or external stimuli. So basically, nervous system is the one who is responsible for sensing, because one of the functions of the nervous system is sensory function, which senses changes or stimuli through its sensory receptors. The second one is the integrative function. This integrative function is achieved by the nervous system, which processes sensory information by analyzing it and making decisions for appropriate responses, which is an activity known as integration. So after sensing or after doing the sensory function, our nervous system also does the integration or the integrative function, in which it analyzes the information it has received from the sensory receptors, and it will now proceed to the motor function, in which the nervous system elicits an appropriate motor response by activating effectors. So if you will observe these functions of the nervous system, you may somehow relate it with the feedback systems we have already discussed on chapter 1 or lesson 1, where there are the receptors, which receives the information or senses the changes or stimuli. Then it gives an input towards the control center. The control centers, which now acts as the integrative function because it analyzes and makes decision for the or analyze situation from the sensory receptors. Then it will produce an output going to the effector, which is now the motor function, that will produce an effect or action to solve the sense stimuli. So now we will proceed to the histology of the nervous tissue. If you will see this picture, you will observe there is a neuron, which is the large blue one, and there are a lot more smaller cells or neural cells around the neuron. And these smaller cells are called neuroglia or glial cells, or some other called it as neuroglial cells. But as well, it is all the same because neuroglia, glial, or neuroglial, it's all the same. So, first is that nervous tissues composed of two types of cells, the neurons and the neuroglia. It may somehow be confusing because if we reminisce our junior high school, we are only introduced to one nerve cell or one nervous cell, which is the neurons. But this time, we are introduced to the neuroglia, which is the other type of nerve cells. So let's start with the neurons. So as we all know, neurons are the basic working unit of the brain and designed to transmit information to other nerve cells, muscle, or gland cells. So these are the parts of the neurons. We have here the dendrites, the bigger branch looking like. They are the receiving or input portions of a neuron and has numerous receptor sites. So, they are located on the head part of the neurons, which are attached to the cell body, or also called as the perikaryon or soma. Well, it is called cell body because it contains nucleus that is surrounded by cytoplasm that also includes typical cellular organelles. And lastly, this tail part, the tail-looking part of the neurons, is called the single axon or axis, which is where the neuron propagates nerve impulses towards another neuron, a muscle fiber, or gland cell. So dendrites are the receiving ones or the receiving part of nerve impulses, while the axon is the part of the neuron which sends impulses to other nerve cell or to other muscle fiber or gland cells. So, in our cell body, there are specific terms that are used in naming the cell body or the parts of the cell body under the nervous cell or the neurons. So, our cell body, which are in the neurons, contains free ribosomes and clusters of rough endoplasmic reticulum, which are now called the cell bodies. While the cytoskeleton, which includes both neurofibrils, a bundle of intermediate filaments that provide cell shape and support, and also microtubules that assist in moving materials between cell body and axon. And the main difference between these neurons is that when our neurons age, it contains lipofuscin, which are clumps of yellowish-brown granules that is product of neuronal lysosomes, which accumulates as the neuron ages. However, this lipofuscin does not seem to harm the neuron, but it is just there as the neuron ages. While on our axon, the axon of the neuron, the long, thin cylindrical projection that often joins the cell body at a cone-shaped elevation is called axon hillock. So, the part of the axon where it connects to the cell body and has a cone shape is the axon hillock, while the part of the axon that is closest to the axon hillock is called initial segment. And the nerve impulses that arise at the junction of the axon hillock and initial segment are called the trigger zone, while the cytoplasm of an axon is called axoplasm and is surrounded by plasma membrane that is also called axolemma. So, these side branches called axon collaterals may branch off typically at right angle to the axon and end by dividing into many fine processes called axon terminals or axon telodendria. So, if you will look at this picture, you will see the detailed parts or more detailed parts of the neurons. So, if you will look at here closely, you will see the axon hillock, which is part of the axon that is connected with the cell body or the soma. And just beside it is you could find the initial segment, and between them is the trigger zone where nerve impulses generates or originates. Then you could see those other parts that are more detailed, just like the myelin sheath, the one that covers the axon, which we will further discuss later on. So next is the synapse, or some other called it as synapse. This synapse or synapse is a site of communication between two neurons or between a neuron and an effector cell. So the tips of some axon terminals swell into bulb-shaped structures that are called synaptic end bulbs. And the string of swollen bumps are called varicosities. Both of the synaptic end bulbs and varicosities contain many tiny membrane-enclosed sacs called synaptic vesicles, which stores the chemical called neurotransmitter. So going back to this picture, if you will see the end part of the axon, you will also see branches similar with the dendrites. However, these branches are called the axon terminals. And at the end, you will see a circular part at the end of axon terminal, circular or something that is swollen or bulb-shaped, which are called the synaptic end bulb. And if you will look closely at that synaptic end bulb, you will find the neurotransmitters because it is there where you could find the synaptic vesicles where neurotransmitters are stored. So in this synapses, there are different transport systems. Specifically, there are two kinds of transport systems. It could be slow axonal transport or fast axonal transport. The first one, the slow axonal transport, travels only 1 to 5 millimeters per day, meaning it conveys axoplasm in one direction only. It is from the cell body to the axon terminals. It also supplies new axoplasm to developing or regenerating axons and replenishes axoplasm in growing and mature axons. While the fast axonal transport has about 200 to 400 millimeters per day. Because they use these proteins that function as motors to move materials, they tend to move faster. And they move materials in both directions. It could be away or towards the cell body. So they are further classified into two, which could be anterograde or retrograde. The anterograde moves in forward direction, in which it moves organelles and synaptic vesicles from the cell body to the axon terminals. However, fast axonal transport could also be retrograde. This retrograde moves in backward direction, in which the membrane vesicles and other cellular materials are moved from the axon terminals to the cell body to be degraded or recycled. So those are the two kinds of transport systems, which also has the anterograde and retrograde under the fast axonal transport. So now we will proceed to the different classifications of neurons. Well, there are two kinds of classification of neurons. First is based on structure, and it could also be based on function. But first, let us discuss the structural classification of the neurons. In the classifications of neurons based on the structure, we have four classifications: the multipolar neurons, bipolar neurons, unipolar neurons, and pseudo-unipolar neurons. So first is the multipolar neurons. They are usually have several dendrites and one axon, and are usually found in the brain and spinal cord as well as all motor neurons. If you will observe the picture of the multipolar neurons, it is called multipolar because it has numerous dendrites and one axon, thus having numerous polarities of the neurons. The second one is the bipolar neurons, which only have one main dendrites and one axon, and they are found in the retina of the eye, inner ear, and the olfactory area or the nasal area. So based on the picture, you will see that there is one dendrites and one axon, giving it only two polarities. That is why it is called bipolar neurons. The third one is the unipolar neurons. Unipolar neurons have dendrites and one axon that are fused together to form a continuous process that emerges from the cell body. So if you will see on the picture, the dendrites and the axon are joined together, thus giving it only one polarity. That is why it is called unipolar neurons. Then lastly, we have the pseudo-unipolar neurons, which begins in the embryo as bipolar neurons and are found in sensory receptors. If you will look at the pseudo-unipolar neurons, it has dendrites and axons that are somehow branch but still together, and they begin in embryo, that is they are called pseudo-unipolar neurons. So after the structural classification, we have the functional classification. Compared to the structural classification, in functional classification, we only have three classifications of neurons. That is the sensory or afferent neurons, the motor or efferent neurons, and the interneurons or association neurons. The sensory or afferent neurons is called afferent because afferent means towards. Since sensory neurons sends information or nerve impulses towards the central nervous system, they are called afferent neurons. They either contain sensory receptors at the distal ends or located just after the sensory receptors, or separate cells, and they are usually unipolar. The second type is motor or efferent neurons, which efferent means away because motor neurons sends nerve impulses from the nerve system or from the central nervous system away from it towards the other nervous tissues, since they convey action potentials away from the central nervous system to effectors in the periphery or peripheral nervous system. So it could be either through cranial or spinal nerves, and they are mostly multipolar in structure. Lastly is the interneurons or association neurons. Well, interneurons or association neurons is the one who is different from the three because it is the only one that is located in the central nervous system. It is between the sensory and motor neurons which integrate incoming sensory information and elicit motor response. And they are usually multipolar in structure. So these interneurons or association neurons are the ones who place the integrative function of the nervous system. Remember the one that we have discussed a while ago, the functions of the nervous system? The sensory function is played by the sensory afferent neurons, while the motor function is played by the motor afferent neurons, while the integrative function is played by the interneurons or association neurons. So if you will look at this diagram, you will see that the sensory neuron and the motor neuron are both could be found under the peripheral nervous system, while the interneuron is the only one that could be found under the central nervous system. And the sensory neuron is connected to the interneuron, and that interneuron is connected with the motor neuron. So now we are finished with the neurons, and we will now proceed to the other type of nerve cell, which is the neuroglia or glial cells. So, this neuroglia actively participates in the activities of nervous tissue. They are smaller than neurons and about 5 to 25 times more numerous. The main difference is that they do not generate or propagate action potentials, but they can multiply and divide in the mature nervous system. Just for trivia, brain tumors derived from glia or glial cells are called gliomas that tend to be highly malignant and grows rapidly. So basically, these neuroglia or glial cells are the nerve cells that supports the neurons or the basic unit cell of the nervous system. So there are a lot of kinds of neuroglia that could be found in the central nervous system and the peripheral nervous system. So first is in the central nervous system. We have the astrocytes. These astrocytes are the star-shaped cells, the largest and most numerous, in which they support and isolate neurons by secreting chemicals that maintain unique selective permeability. In the embryo, astrocytes secrete chemicals that regulate growth, migration, and interconnection among neurons. So, in short, these astrocytes are the ones who supports the basic activities or the daily activities of the neurons. And there are two types of it, which are the protoplasmic astrocytes and the fibrous astrocytes. First is the protoplasmic astrocytes, which are short branching and smaller, that could be found in gray matter, while fibrous astrocytes are long unbranched processes that could be found in white matter. The second type of neuroglia in CNS is the oligodendrocytes. These oligodendrocytes resemble astrocytes but are smaller and contain fewer processes. They are responsible for forming and maintaining the myelin sheath around CNS axons. So oligodendrocytes may somehow look like astrocytes, but their function differs from the astrocytes because these oligodendrocytes only maintains and makes myelin sheath on the CNS axons. The third kind is the microglia. These microglia are small cells with slender processes that give off numerous spine-like projections. So they function as phagocytes which remove cellular debris formed during normal development of the nervous system and phagocytize microbes and damage nervous tissue. So basically, microglia functions as macrophages or other phagocytes in the nervous system on the central nervous system. So, they wander around the central nervous system and they phagocytes the foreign substances, the microbes, and other damaged nervous tissue. Then we have here the last type of neuroglia or glial cells in central nervous system, which are called ependymal cells. These ependymal cells could vary from cuboidal to columnar cells, which are arranged in single layer that possesses microvilli and cilia. If you could recall, microvilli and cilia are parts of the cell which helps in the motor function of the cell itself. So they line the ventricles of the brain and the central canal of the spinal cord. They basically produce, possibly monitor, and assist in the circulation of the cerebrospinal fluid. So that cerebrospinal fluid is the fluid that we could found within the brain and the spinal cord. So if you will look at this picture, you will see the different types of the neuroglia or glial cells that could be found in the central nervous system. Here is the protoplasmic astrocyte, which is the larger kind of astrocyte, which are the long branching, while the fibrous astrocytes are the smaller and short branching. Then we have here the oligodendrocytes that is attached within the myelin sheath since it monitors and produces myelin sheath for the central nervous system axons. Then we have here the microglial cells. The two microglial cells, one is attached on the dendrites and one is attached on the ependymal cells, since it wanders off around the central nervous system as it looks for foreign substances, cellular debris, and unused or dead nervous tissues to be phagocytized. So we have here the ependymal cells just below the neurons. Those are arranged there to monitor and maintain the circulation of the cerebrospinal fluid. So now we will proceed to the neuroglia of the peripheral nervous system. So in peripheral nervous system, there are only two kinds of neuroglia or glial cells. And these are the Schwann cells and the satellite cells. The Schwann cells are like oligodendrocytes, meaning they encircle PNS axons and form myelin sheath around axons. So just like oligodendrocytes, they are the ones who are responsible for making the myelin sheath of the axons in the peripheral nervous system. So they also participate in axon regeneration, which is more easily accomplished in the PNS than in the CNS. So the question is, why is it much easier to undergo axon regeneration in the PNS than in the CNS? So this time, three persons who could answer, or three persons who could first answer this question, who could answer correctly this question by sending me a PM, would receive a 100 peso GCash. 100 peso through GCash. So the question is, why is axon regeneration much more easily accomplished in the PNS or peripheral nervous system than in the central nervous system? So the first three persons who could answer correctly would receive 100 peso through GCash. So just PM me the answer. So now we will proceed with the satellite cells. These satellite cells are flat cells that surround the cell bodies of neurons of PNS ganglia. They regulate the exchange of materials between neuronal cell bodies and interstitial fluid. So this satellite cells surrounds the cell bodies. This is much more different because um, it doesn't have similar cells within the central nervous system because it's only found in the peripheral nervous system. So they basically surround the cell bodies and they regulate the exchange of materials and waste between the cell bodies and the interstitial fluid. So if you will look at this picture, you will see the two kinds of neuroglia in the PNS. The first one in here, the letter A, you could find the Schwann cell in a myelinated axon, while in the second one is you could find the Schwann cell even in unmyelinated axon, meaning those axons that doesn't have myelin sheath. Then lastly, you will find the satellite cell that is attached to the cell body. That is the one who is responsible for the exchange of materials between the cell body and the interstitial fluid. So, a while ago, we have stated or we have heard the myelin sheath for how many times? So this time, let us discuss what is myelin sheath. So this sheath is a multi-layered lipid and protein covering which electrically insulates the axon of a neuron and increases the speed of nerve impulse conduction. So basically, this myelin sheath is the one who helps on the speed of the nerve impulses, on the travel of the nerve impulses. So, axons could be either myelinated or unmyelinated, meaning it could have myelin sheath or it could does not have any myelin sheath at all. So, Schwann cells, which are in the PNS and the oligodendrocytes in the CNS, produces these myelin sheaths. And the amount of myelin increases from birth to maturity, and its presence greatly increases the speed of nerve impulse conduction. So therefore, myelinated axons conveys much faster nerve impulses than the unmyelinated axons. Since unmyelinated axons doesn't have myelin sheath, it doesn't have a part that helps in the increase of speed of nerve impulses conduction. So in this picture, you could see how myelin sheath develop or form through stages. So here in letter A, you could see that from Schwann cell, it releases or produces a myelin sheath, and further on, it moved circularly or roundly as it covers the axon completely. If you will observe, it is somehow similar with how the osteons or haversian systems in our bones are fully completed or fully formed. Because as it forms or as it rounds around the axon, the myelin sheath has formed layers and layers of myelin sheath around the axon. And the second picture is the unmyelinated axon, which doesn't have myelin sheaths at all. So what is white and gray matter? A while ago, when we are discussing the two kinds of astrocytes, where the fibrous astrocytes could be found in white matter, and the other one could be found in gray matter. What is the main meaning of white and gray matter? Well, aside from its color, the difference of its color, the gray matter contains neuronal cell bodies, dendrites, unmyelinated axons, axon terminals, and neuroglia. While white matter is composed primarily of myelinated axons, the whitish color of myelin gives white matter its name. Well, basically, if you will look at our brain, it is not specifically or literally white and gray matter. It just shows that the white matter, which is lighter in color, shows that that part has axons that are myelinated, while the gray matter, which are the darker sides or darker parts of the brain, has unmyelinated axons. because since myelin sheets are looking or looks like white, those axons that has myelin sheets appear to be on the white matter, while those axons or neurons that doesn't have myelin sheets or unmyelinated are on the gray matter or appears to be darker. So it appears grayish rather than white because the nisal bodies which are the endoplasmic reticulum and the ribosomes impart a gray color, and there is a little or no myelin in these areas. So, if you will look at this, the border of the brain, most of the border of the brain and the middle part are the gray matter, while the middle part, the center part of the brain consists of the white matter. So now we will proceed with the electrical signals in neurons. So as we all know, our neurons are very excitable cells, and they generate or sends electrical signals to generate action or to send information. These neurons communicate with one another using two types of electrical signals. It could be graded potentials, which are used for short distance communication, and we have the action potential, which allows communication over long distances within the body. So, the production of this potential, could be graded or action potential, are dependent on the two basic features of the plasma membrane. Meaning, these two features of plasma membrane are the two factors that affects the production of the graded potential and the action potential. And these are the resting membrane potential and the presence of the ion channels. So, just a few definition. Membrane potential is an electrical potential difference or voltage across the membrane. This voltage is termed the resting membrane potential. So, if you will look at this diagram or picture, you will see how graded potentials and action potentials in both nerve and muscle happens as we write on the paper through a pen. So, starting from our fingertips, we have there our sensory receptors. And through our sensory receptors, we sense the pen that we hold. So, from the main sensory receptor going to the next neuron, there happens the graded potential. And from that neuron going to the sensory neuron happens to be a nerve action potential, since it is a long distance communication, because from that sensory receptor going to the brain or to the interneuron, it is such a long distance, that is why it undergoes a nerve action potential. However, before going to the brain, as it as it transfers to the interneuron from the sensory neuron, it undergoes graded potential, then goes back in doing the nerve action potential. So, as the information travels to the cerebral cortex, the specific part of our brain, it goes or it travels through nerve action potential. And when it reaches that specific part of the brain, the output that the brain has produced undergoes a graded potential and will further travel through nerve action potential. So from there, it will travel to the spinal cord, and here in the spinal cord, it will be transferred into another neuron through graded potential. Then it will travel again distantly through nerve action potential until it reach the neuromuscular junction. If you would remember, when we have studied the muscular system, we have discussed the neuromuscular junction, in which These are motor neurons that are, or motor end plate that are attached to the skeletal muscles. And from that, the nerve action potential has been transformed into muscle action potential, which generates the muscle to contract and produces an action. So that is the whole process of the nerve impulses and how graded potential and action potentials are produced alternatively. So, as I have said a while ago, graded potentials and action potentials are both dependent on these two factors, the ion channels and the resting membrane potential. So first, let us discuss the ion channels. So, ion channels are open and closed due to the presence of gates. The gate is a part of the channel protein that can seal the channel pore shut or move aside to open the pore. If we would remember when we have studied the different transport system or active transport system within the plasma membrane, we have already discussed different kinds of gated channels or ion channels. This time, in nervous system or in our neurons, there are four kinds of ion channels. The first one is the Lick channels, which are gated channels that randomly open and close and are found nearly in all cells. So, these Lick channels just randomly opens and close. It does not need a specific ion, specific ATP, specific chemical substance to open or close as it randomly opens and closes. So the next one is the ligand-gated channel, which are gated channels that open in response to binding of ligand or chemical stimulus that are found in dendrites of some sensory neurons and dendrites and cell bodies of interneurons and motor neurons. So if you remember the receptor-mediated transport or receptor-mediated channel when we have discussed the transports or the different transports under the plasma membrane, it is somehow similar with these ligand-gated channels because a specific ligand or specific chemical attaches to the receptor, it will automatically opens and transport that specific chemical substance inwards the cellular or plasma membrane. So, this ligand-gated channels undergoes that process in which it has a receptor, and once a ligand is attached to the receptor, it would be transported inwards the plasma membrane. The third type is the mechanically gated channels. So these gated channels that open in response to mechanical stimulus. They are found in dendrites of some sensory neurons such as touch receptors, pressure receptors, and some pain receptors. So since they are mechanically gated channels, meaning they open and close in response to mechanical pressure or mechanical stimulus, they are only found within the receptors such as the touch receptors, pressure receptors, and pain receptors. And the last kind of ion channel is the voltage-gated channels, in which gated channels that open in response to voltage stimulus, meaning this is the change in membrane potential. And they are found in axons of all types of neurons. So if you will look at this diagram, you will also see the summary of these ion channels. The first one is the Lick channel, which randomly opens and closes. The ligand-gated channel, which opens and closes through chemical stimulus. And then we have the mechanically gated channels that opens through mechanical stimulus. And lastly is the voltage gated channel that opens through the change in membrane potential. So if you will observe from the negative 70 millivolts, when the voltage has changed into negative 50 millivolts, then the voltage gated channel has opened. So the negative 50 millivolts could go back into the resting membrane potential, which is the negative 70 millivolts. So now we will proceed to the second factor that affects the production of graded and action potentials. So this is the resting membrane potential. So it exists because of a small buildup of negative ions in the cytosol along the inside of the membrane and an equal buildup of positive ions in the extracellular fluid. Meaning, resting membrane potential is where there is the presence of polarity across the membrane, in which the cytosol or the extracellular fluid has a more negative charge, while the extracellular fluid has a more positive charge. The greater the difference in charge across the membrane is the larger the membrane potential. So in neurons, the resting membrane potential ranges from 40 to 90 millivolts, in which a typical value is around 70 millivolts. So the resting membrane potential arises from three major factors. It could be the unequal distribution of ions in the ECF and cytosol, the inability of most anions to leave the cell, and lastly is the electrogenic nature of the sodium-potassium ATPases. If you will look at this picture, you will see that the cytosol and the extracellular fluid both has the equal numbers of positive and negative charges. However, the one that are near with the plasma membrane has the polarity in which in the extracellular fluid has a more positive charge while in the cytosol has a more negative charge, which is why it is called the resting membrane potential. That is an electrical potential difference across the plasma membrane. So, when we measure this resting membrane potential through a voltmeter, we will measure it around negative 70 millivolts, which is the typical measurement of the resting membrane potential. In this photo, you could see that these channels or the ion channels are the ones who help to maintain the resting membrane potential. So, let us proceed with the graded potentials. Graded potentials are small deviation from resting membrane potential that makes membrane either more polarized or less polarized. So, it could be hyperpolarizing graded potential or depolarizing graded potential. In hyperpolarizing graded potential, its response makes the membrane more polarized, meaning the inside or the cytosol would be more negative. That is why from the word itself, hyperpolarizing, it makes the plasma membrane it makes the polarity in the plasma membrane much higher because it gives more negative charge in the inside of the cell or in the cytosol. While in depolarizing graded potential, its response makes the membrane less polarized, meaning the inside would be less negative or more positive. That is why from the word itself also, it is depolarizing, meaning making it more less polar. So this mode of travel by which graded potential die out as they spread along the membrane is known as decremental conduction. It can be stronger and last longer by summating with other graded potentials, in which summation is the process by which graded potentials add together. So, since we have discussed that graded potentials are only known for short distance communication, it can somehow travel a little bit longer through the process of summation, in which this graded potential add with each other or add together to travel at least much longer than the normal ones. So graded potentials have different names. It depends on which type of stimulus causes them and where they occur. For example, the postsynaptic potential, the receptor potentials, and the generator potentials. So if you will look at this picture, you will see that in the letter A, which is the hyperpolarizing graded potential, where the polarity on the plasma membrane is much more higher, you could see that the millivolts has been -80 or moved towards less negative or more negative because the polarity has been higher than the normal. However, in the depolarizing graded potential, the resting membrane potential has moved in a lower or higher millivolts going towards the -60 since it became less negative or more positive. So, let us now talk about the generation of action potentials. So, this action potential or AP or impulse is a sequence of rapidly occurring events that decrease and reverse the membrane potential and then eventually restore it to the resting stage. So there are two main phases of the action potentials. We have depolarizing phase and repolarizing phase. Unlike the graded potential, which has the hyperpolarizing and depolarizing, the action potential has depolarizing and repolarizing phase. First, in the depolarizing phase, the negative membrane potential becomes less negative, which reaches zero and then becomes positive. So, meaning it is similar with the graded potential, the depolarizing phase or depolarizing graded potential, where the resting membrane potential became less negative, which become more positive. So, in repolarizing phase, the membrane potential is restored to the resting state of 70 millivolts. So, in repolarizing phase, it is just about going back to the resting membrane potential, which is about the -70 millivolts. So, there is what we call the after hyperpolarizing phase. This after hyperpolarizing phase is during which the membrane potential temporarily becomes more negative than the resting level. Meaning, this is the point or phase where the resting membrane potential became the most negative from the rest of the phase. So in this photo, you could see that in the purple one, it is the resting membrane potential. The blue one, it is where the stimulus has been made or has been encountered. In the green one, the voltage-gated sodium ion channel are open. And on the yellow or the lighter orange is where the voltage-gated potassium ion channels became open. And on the other hand, the sodium ion channels became inactivated. And in the orange part is where the voltage-gated potassium ion channels are still open and the sodium ion channels are on the resting stage. So, under the resting membrane potential, we could see that the curve is still flat because it is still on the resting membrane potential. However, when it have encountered a stimulus, it started to depolarize and reach the threshold. And once it reached the threshold, the voltage-gated sodium ion channel started to open, which makes the polarity much more depolarized, in which reach the depolarizing phase or the peak of depolarizing phase. And once the voltage-gated potassium channels have also opened and the sodium ion channels started to inactivate, it now started to repolarize or goes back to the original resting membrane potential, which is -70 millivolts. So as the curve goes down, it reached the after hyperpolarizing phase, which is the phase where the resting membrane potential has reached its most negative than the rest of the phase or levels. Then after reaching that after hyperpolarizing phase, it will go back to its normal resting membrane potential that is just about negative 70 millivolts. So that is how action potentials work. Moving on in generation of action potentials, an action potential occurs in the membrane of the axon of a neuron when the polarization reaches a certain level termed the threshold, which is about 55 millivolts in many neurons. So to generate an action potential, we should reach the threshold, which is about 55 millivolts. And an action potential will not occur in response to a sub-threshold stimulus, which is a weak depolarization that cannot bring the membrane potential to threshold. So this sub-threshold stimulus is much weaker than the normal threshold stimulus, because to generate an action potential, we should reach the threshold, which is 55 millivolts. That is why we should remember the 55 millivolts. Potential will occur in response to a threshold stimulus, a stimulus that is just strong enough to depolarize the membrane to threshold. Yan. Because when we reach the threshold stimulus, we are now able to produce or generate an action potential, because that threshold is strong enough to depolarize the membrane, because to produce an action potential, we should depolarize the resting membrane potential, meaning make it less negative. Lastly, several action potentials will form in response to a suprathreshold stimulus, a stimulus that is strong enough to depolarize the membrane above threshold. Ayan, we have the suprathreshold stimulus. This threshold or this depolarization is the one that reach above the normal threshold. Meaning, since it is much stronger or much higher than the normal threshold, it could generate several action potentials. Because action potential either occurs completely or does not occur at all. This characteristic is known as the all or none principle. That is why when it does not reach the threshold stimulus, or it is just around the sub-threshold stimulus, it will never generate an action potential, because action potentials are known as the all or none principle, which could only produce or not produce an action potential. So in this diagram, you could see how sub-threshold stimulus, threshold stimulus, and supra-threshold stimulus results. So in sub-threshold stimulus, you could see that it depolarizes only a little amount, only around negative 60 millivolts from negative 70, while threshold stimulus, which is the requirement to produce an action potential, has reached at about 30 millivolts, and suprathreshold stimulus has produced about 3 action potentials, because going back to its definition, suprathreshold stimulus are above the threshold stimulus, which is capable of producing several action potentials. However, in action potentials, there are what we call refractory period. This is the period of time after an action potential begins, during which an excitable cell cannot generate another action potential in response to a normal threshold stimulus. So, refractory period, this is the period where action potentials cannot be produced again after generating one. So, there are two kinds. First is the absolute refractory period and the relative refractory period. The absolute refractory period, even a very strong stimulus cannot initiate a second action potential, while relative refractory period is the time which a second action potential can be initiated, but only by a larger than normal stimulus. So when we say absolute, meaning it is that is already that when you produce an action potential, that's it. You can produce another action potential because you have already reached the absolute refractory period. However, the relative refractory period could produce until second action potential just by using or when there is a suprathreshold stimulus or much larger and stronger stimulus than the normal threshold. But after that, second action potential, you could no longer produce another action potential. Next is the propagation of action potentials. So, action potential keeps its strength as it spreads along the membrane. This mode of conduction is called propagation, and it depends on positive feedback. So, there are two kinds, the continuous conduction and the saltatory conduction. The continuous conduction involves step-by-step depolarization and repolarization of each adjacent segment of the plasma membrane, while saltatory conduction is the special mode that occurs along myelinated axons because of the uneven distribution of voltage-gated channels. So, under the saltatory conduction, the flow of current across the membrane only at the nodes of Ranvier has two consequences. It is either the action potential appears to leap from node to node as each nodal area depolarizes to threshold, which travels much faster, or the opening of a smaller number of channels only at the nodes rather than many channels in each adjacent segment of membrane, which represents more energy efficient mode of conduction. So based on this diagram, you could see that continuous conduction is happening or manifesting only in the unmyelinated axons because since it doesn't have myelin sheaths, it generates continuous conduction. While in the saltatory conduction, it only happens in the myelinated axons since there is the presence of myelin sheaths and nodes of Ranvier. Since there are the nodes of Ranvier, the generation of nerve impulses or the process or conduction of the nerve impulses happens to leap or undergoing or appears to leap from node to node since there are the presence of the myelin sheath. There are also factors that affects the speed of propagation. The amount of myelination, the axon diameter, and the temperature. So first is the amount of myelination. Of course, since myelin sheaths help to speed up or to increase the speed of propagation, therefore, myelinated axons tend to have faster propagation rather than the unmyelinated axons. The second one is axon diameter. The larger the diameter of the axon, it can propagate action potentials faster because there are larger surfaces unlike on the smaller diameter of axons. Then lastly, we have the temperature in which axons propagate action potentials at lower speeds when cooled. Therefore, if there is a higher temperature, then axons propagate action potentials at a faster rate. So, there are also signal transmission at synapses. So, again, when we say synapse or synapse, it is a region where communication occurs between two neurons or between a neuron or an effector cell. So, there are two kinds of neuron when we are talking about synapses. These are the presynaptic neuron and the postsynaptic neuron. The presynaptic neuron are the nerve cells that carries nerve impulses towards a synapse. Thus, it is the cell that sends signal, meaning it is the neuron where synapse or nerve impulses originated, while the postsynaptic neuron is the cell or nerve cell that receives the signal coming from the presynaptic neuron. It carries a nerve impulse away from a synapse or an effector cell that responds to the impulse at the synapse. Most synapses between neurons could be axodendritic, meaning these are from axon to dendrites, while others are axosomatic, meaning from axon to cell body, and axoaxonic, meaning from axon to axon. So when we look at this picture, the large neuron here is a postsynaptic neuron. So in this picture, you could see the three kinds of synapses. First is the axoaxonic, which is axon connecting to the axon, while there is the axodendritic, in which axon connects with the dendrites, while axosomatic is the one which connects the axon towards the cell body or the soma. So those are the three kinds of synapses. And there are also two kinds of synapses depending on its potentials. So it could be either electrical synapses or chemical synapses. First is the electrical synapses. These are action potentials that conducts directly between the plasma membrane of adjacent neurons through structures called gap junctions. So I hope you could still remember gap junctions, which we have discussed on the first lessons around the tissues. And electrical synapses have two main advantages. It is much faster, and of course it is more synchronized. Why? Because first, it is much faster because action potential passes directly from the presynaptic left or presynaptic cell to the postsynaptic cell, while it is synchronization because large number of neurons or muscle fibers can produce action potentials in unison if they are connected by gap junctions. On the other hand, we have the chemical synapses where presynaptic neuron releases neurotransmitters that diffuses through fluid in synaptic cleft. Postsynaptic neuron receives and in turn produces postsynaptic potential, a type of graded potential. There is the presence of synaptic delay, which is about 0.5 millisecond, which is the reason that chemical synapses relay signals more slowly than electrical synapses. So based on this picture, you could see the process of how chemical synapses happen. So from the nerve impulses that travels in the presynaptic neuron, it goes here at the end of the axon terminal in synaptic vesicles where it produces neurotransmitters and goes within this synaptic cleft, which is a space between the axon terminals, synaptic bulb, and the postsynaptic neuron, because the axon and the dendrites does not meet specifically because there is a small gap called synaptic gap or synaptic cleft, and from that neurotransmitters are received through the neurotransmitter receptors. So neurotransmitter causes either an excitatory or an inhibitory graded potential. The APSP or the excitatory postsynaptic potential, neurotransmitter that causes depolarization of the postsynaptic membrane, is excitatory because it brings the membrane closer to threshold, while hyperpolarizing postsynaptic potential or inhibitory postsynaptic potential or IPSP is the generation of action potential that is more difficult than usual because membrane potential becomes more negative inside and thus even farther from the threshold than its resting state. So, those are the two kinds of postsynaptic potentials that could be generated in the chemical synapses. And upon transferring neurotransmitters, there are different kinds of neurotransmitter receptors that could be found in the postsynaptic neuron. These neurotransmitters released from a presynaptic neuron bind to neurotransmitter receptors in the plasma membrane of a postsynaptic cells. So it could be either ionotropic receptors or metabotropic receptors. The ionotropic receptors contains neurotransmitter binding site and ion channels, meaning the ion channels and the binding sites are same protein, components of the same protein, and shown as a 1, while metabotropic receptors contains neurotransmitter binding site and ion channel that are separated from each other, but they are coupled with each other. So if you will look at this picture, you will understand it more clearly because letter A and letter B shows a ionotropic receptor in which binding site and ion channel are one with each other, while in metabotropic receptor, the ion channel and the binding site is different from one another, but they are coupled or they are partner with each other. So there are different removal of neurotransmitter because once the neurotransmitter have sent the nerve impulses or electrical signals, it should go back or send back to the presynaptic neuron that sent that neurotransmitters. So the removal of the neurotransmitter from the synaptic cleft is essential for normal synaptic function. So there are three forms of removing neurotransmitter. First is the diffusion, the enzymatic degradation, and the uptake by cells. Diffusion is where some of the released neurotransmitter molecules diffuse away from the synaptic cleft. So after sending the nerve impulses or electrical signals, it diffuses away. The second type is the enzymatic degradation, meaning certain neurotransmitters are inactivated through enzymatic degradation. So there are enzymes that are produced to inactivate this neurotransmitter and to be removed in the synaptic cleft. Lastly, there is the uptake by cells in which many neurotransmitters are actively transported back into the neuron that released them, meaning they are undergoing a process of reuptake in which they are sent back to the presynaptic cell or presynaptic neuron that sent those neurotransmitters. So there are two kinds of summation of neurotransmitters or postsynaptic potentials. These are the spatial summation and temporal summation. When we say spatial summation, these are summation of postsynaptic potentials in response to stimuli that occur at different locations in the membrane of a postsynaptic cell at the same time. While temporal summation of postsynaptic potential in response to stimuli that occur at the same location in the membrane of the postsynaptic cell but at different times. So the main difference is that spatial summation is the sum of postsynaptic potentials where it is on the same time, happening on the same time, but in different locations. But in temporal summation, it is the same location but different times. So those are the main difference of those two kinds of summation. And this is a diagram that explains those. This one is a spatial summation and this one is a temporal summation. So the sum of all the excitatory and inhibitory effects at any given time determines the effect of the postsynaptic neuron, which may respond in the following ways. It could be EPSP, or excitatory postsynaptic potential, nerve impulses, or IPSP, or inhibitory postsynaptic potential. So if the total excitatory effects are greater than the total inhibitory effects, but less than the threshold level, then it is EPSP. However, nerve impulses is reached when excitatory is much more higher or greater than the inhibitory effects and the threshold is reached. However, it would be inhibitory or IPSP if inhibitory effects is much more higher than the excitatory. So those are the three kinds of the sum of all excitatory and inhibitory effects. So this is a diagram in which there is the postsynaptic neuron that is in contact with different other presynaptic neurons that produces either EPSP, IPSP, and such. Lastly is the neural circuits. So neural circuits are functional groups of neurons that processes specific types of information. The simple series circuit is a presynaptic neuron that stimulates a single postsynaptic neuron. So there are four kinds of that neural circuits. First is the diverging circuit, the converging circuit, the reverberating circuit, and the parallel after discharge circuit. The diverging circuit is where the nerve impulse from a single presynaptic neuron causes stimulation of increasing numbers of cells along the circuit. In short, in diverging circuit, there are a lot of presynaptic neuron, or there is a single presynaptic neuron that goes into a lot of postsynaptic neuron. While converging circuit is where the postsynaptic neuron receives nerve impulses from several different sources. So this is in comparison or opposing with the diverging circuit, because in converging circuit there are a lot of presynaptic neuron, and there is only one postsynaptic neuron. While in reverberating circuit, the incoming impulse stimulates with first neuron, then to the second, then to the third, and so on. So therefore, it flows in a chronological way in which from the first one, it travels to the second, to the third, and so on and so forth. Lastly is the parallel after discharge circuit, in which single presynaptic cell stimulates a group of neurons, each of which synapses with a common postsynaptic cell. So if you will look at this picture, you will understand it more, because first, in the diverging circuit, you could see that it started from a single presynaptic neuron, then goes with several or numerous postsynaptic neuron, meaning it diverges from one, it scattered into numerous other postsynaptic neuron. While in converging circuit, it started from numerous or several presynaptic neuron going to a single postsynaptic neuron, meaning all of the information or all of the synapses converges to one postsynaptic neuron. While in reverberating circuit, it happens to be chronological in order, as starting from the first neuron, then to the second neuron, then to the third neuron, then fourth, fifth, what, and so on. While in the parallel after discharge circuit, you will see that it started with a presynaptic neuron, then it travels in a group of neurons until it reaches a single postsynaptic neuron. That is why it is called parallel after discharge circuit. And that's it. That's my whole report, and we will now proceed with the next topic.